Biotechnology principles and processes. This is the chapter that we will be starting today. It will be a lecture series in which the first lecture will deal with mainly the introduction on biotechnology and the basic principle as also the basic process involved. And in the subsequent lectures, I'll be talking about the different steps involved in details. So let us start with it. Now, what is biotechnology? Biotechnology means the use of microbes, plants, or animal cells, or any of their part, any of the components, which will help in generating products, which will help in creating some products. Okay, creating some products which can be a protein, which can be a carbohydrate, which can be a fat, uh, which that will help human beings. It can be in the form of an antibiotic. Okay, it can be in the form of an antibiotic, it can be in the form of a hormone, it can be in the form of an enzyme. So those components that will help human beings. So biotechnology involves that. How do we use, how do we create those products? Now, sometimes it so happens that one organism, say organism A, is producing a product, but product is very good, but quantity is very less. There is another organism B which can also be made to produce the product and in huge quantities. So can't we use organism B to do that? So what will be done through biotechnology? What we can do is we can transfer the gene from A to B organism, thereby make B capable of producing the product in huge quantities, which will be of benefit to human beings. So this way, biotechnology can be used to produce products. Now, for the first time in the year 1972, such a new DNA, novel DNA, recombinant DNA was created by the scientists Stanley Cohen and Herbert Boyer. What did they do? The first recombinant DNA was created in Escherichia coli. How did they do? They had an organism which had the antibiotic resistance gene, which could produce an antibiotic resistant gene. So what did they do? They took out that gene, they placed it in the plasmid of Salmonella typhimurium. Then that plasmid, if you can recall, plasmid is the extra chromosomal DNA. Plasmid is the extra chromosomal DNA, circular DNA you find in bacteria. That plasmid is then transferred to Escherichia coli. So finally, what will happen? Escherichia coli will produce the product. So that was how the first recombinant DNA was created by Cohen and Bayer. Now, what are the basic principles involved in biotechnology? That is genetic engineering and maintenance of sterile environment. Now, maintenance of sterile environment means to maintain aseptic conditions free from any contamination. So wherever we are walking, wherever we are carrying out the processes of these particular to create something, then that entire environment has to be made germ free, has to be kept germ free. So different steps can be taken up for that. Alcohol, cleaning with alcohol, UV radiations, autoclaves, so different steaming, hot air ovens, so different processes are involved, but ultimately the aim will be to maintain a sterile environment. Then the process is carried out through genetic engineering. Now, the technique of genetic engineering involved in biotechnology. 3 steps: creation of recombinant DNA, use of gene cloning, and gene transfer. So these are different steps or measures in genetic engineering. It involves to create a new DNA, recombinant DNA, as also to use gene cloning. Gene cloning, now first, what is recombinant DNA? Recombination. So the term itself is self-explanatory, to create a new DNA by combining two different DNAs. Use of gene cloning. Gene cloning means cloning. Cloning means creating new copies, replicas. So even this technique has to be used, and then gene transfer. So what do we do? What is used? Gene from one organism is transferred into another organism. Now different steps are involved in all of this, but these are the basic techniques of genetic engineering involved in biotechnology. Now the three basic steps in genetically modifying an organism. What are the three basic steps that are involved in modifying an organism to create a new organism? First is, we have to identify a DNA with desirable genes. So what is done? First, we have to recognize an organism. First, of course, what do we want? Suppose, for example, we want a particular antibiotic, let us say, okay, against a germ. Some germ is existing, some fungus is existing. So what do we want? We want an antibiotic against it. So what we'll have to look? We'll have to look for other organism who is capable of producing an antibiotic and killing that fungus. So we'll have to identify, okay, that organism. Okay, it is producing an antibiotic. Then we will have to identify the DNA inside it, okay, with the desirable genes, that it has the desirable gene. Now what is the meaning of desirable gene? Desirable gene means the gene which is capable of producing our required product, okay, through transcription and translation. So we'll have to identify that DNA. So here just a picture to show that. Then next is, after that what will be done? That desirable, that particular gene will be cut out. Okay, here though scissor is shown, it doesn't mean it is cut out with a scissor. Here we use molecular scissors, which are some enzymes. So applying the enzyme to the DNA, we can cut out that particular gene that we want. After we cut out the gene, what will be done? It will be introduced into the host. Now who is the host? Host is the cell where we want the product to be produced. That is the host. Gene was present elsewhere. Now we want some other organism to produce the same product. So host is that organism. Now what will be done? That cut out DNA will now be transferred into the host cell via some other means. Via means those are vectors. Vectors will be used. We will talk about that. Here we are just talking about the steps. Then after that, once we have introduced it, the most important, very significant will be maintenance of introduced DNA in the host and transfer of the DNA to its progeny. So that introduced DNA has to be maintained. It is not that we just introduce it and let it go on that way. No, we have to maintain it. That has to be maintained. That means it should be such that the introduced DNA remains in the host till we want the product to be produced. As also it has to be transferred to the progeny. As you can see here, the picture that is shown here, the picture that is shown here, as you can see, okay, this is the transform, the host cell. Now it is dividing. As it divides, this red one is the new DNA. Now the new DNA is getting transferred even to the new progenies, the new offspring. So it has to be transferred. Only then we can say that we are capable of maintaining it in the new organism. So again, once more here, basic steps involved in the process. Isolation of genomic DNA, here the first step which I was talking about. So it involves again sub steps. So isolation of the DNA, fragmenting the DNA, that means cutting out, as I told you, cutting out with the help of enzyme, fragmenting the DNA, screening the DNA fragments. That means we have to screen out. After cutting, we have to find out, we have to exactly detect which is the one that we want. Then insertion of the DNA in a vector. So vector will be the intermediate which will help in transferring the DNA into the host. Then introducing the DNA in the host, and finally growing those host cells, and then transformation of the host cell. So what will happen? The host cell will be transformed, will be generating a new product which it was not generating earlier. So these are the three basic steps involved in genetically modifying an organism. Now, what are the tools that are used? What are the tools that are used? There are three main tools. Restriction endonuclease enzyme. Now this is the enzyme that will help in cutting the DNA. It is also known as molecular scissors. It will be used to cut out the desired DNA. Secondly, cloning vectors. Vectors are those which will help in transferring the required DNA to the host cell, to the second organism, from the first to the second organism. From the first, we take out the DNA, cut our required DNA fragment, required gene with the help of the enzyme. Then that required DNA is then inserted into vectors like plasmids, which are extra chromosomal self-replicating DNAs. It is transferred, and then the vectors will be used, or vectors will help in transferring the required DNA into the host. The host we are calling as competent. Competent means a host which is which will be capable of taking up the new DNA. That is called as competent host. So these are the three tools of most important tools of biotechnology. Details we will be learning. Now the processes of recombinant DNA technology. Now the different processes of recombinant DNA technology, the steps, okay, the steps in details. First is, this is some repetition of course of whatever I have talked about little while ago. So here, but little more in details. Isolation of the DNA and plasmid. So first what will be done? The DNA will be isolated. Okay, the DNA will be isolated as well as the plasmid. The vector will be isolated using the same type of digestive enzyme. The same type of digestive enzyme will be used, and thereby what will happen? We will isolate out the DNA as well as the plasmid that we want, or that is carrying the gene of our interest, and plasmid for the vector. After which, cutting fragmentation of DNA at specific location. Specific location means that part of DNA that we want will be cut out using restriction enzyme. We will cut it out, fragment it out, okay, and then we will separate out with the help of gel electrophoresis. We will separate out the entire broken down fragments of DNA with the help of gel electrophoresis and create multiple copies of the fragmented DNA using a technique called as polymerase chain reaction. Then after which, the cut out DNA, we have multiple copies of it now, so we need not go back to the first step again and again. We need not repeat the first step. So repeatedly we can carry out our experiment because we already have multiple copies using polymerase chain reaction. So those multiple copies will now be ligated. Ligated means joined into a vector. After which, the recombinant DNA, now we have created a recombinant DNA because we have joined it with a vector DNA. So now what will happen? The recombinant DNA is then finally transferred into the host cell using DNA ligase. So DNA ligase will be used here, and then we will transfer it. Then once the host cell has got the foreign DNA from here, okay, along with the vector, it has got it. Then finally, what is it capable of now? It is capable of producing the product. The product can be made to produce in large scales using bioreactors. So here the pictorial depiction of the same these steps here. You can see here an example is taken as Escherichia coli bacteria. So what? The plasmid is used here. Plasmid which will be used as the vector. Okay, now this is a cell with DNA containing gene of interest. This has a particular fragment of gene which we want, okay, which is producing some product desired by us. So what will be done? That DNA is first isolated, okay, using different enzymes. It will be used. Everything is digested out. The cell wall, the cell membrane, the cell organelles, everything will be digested out, leaving behind only the DNA. Only the DNA will be left out, and that DNA, that is isolate the DNA, here this is the DNA that is isolated. Here we have the gene of interest. Okay, similarly even the plasmid is isolated in this manner. Parallelly, separately, this will be carried out. The plasmid is cut with an enzyme. Okay, here this is a circular plasmid. It is cut with an enzyme. The same enzyme will be used to cut even the gene of our interest. Now here we have this gene. This is the gene that will actually produce our product. So what will be done? The gene will now be placed within the plasmid here. Combine target fragment and plasmid DNA. So what is done here? Upon the plasmid, this gene is placed here. The rest of the portion of DNA is discarded. We do not want it. We want only a fragment of the DNA that is the gene of our interest. Then finally, what will happen? This plasmid is complete now. Here a portion was removed. That portion is replaced with our gene of interest, and then it is placed here, joined here. Finally this plasmid, this vector is ready. We will call it as the vector. It will be placed here, recombinant DNA. This is a recombinant DNA. Now it will be placed into the host cell. It will be inserted into a host cell. So of course, here what do we have to use? We have to use an enzyme, DNA ligase, that will help in joining our gene of interest. So finally, it will be transferred into the host cell. So the host cell will then now get the gene that we want it to produce. So it will produce in large quantities. And how can it produce in large quantities? Inside bioreactors. Now, this was just an introduction on the different processes of recombinant DNA technology. Of course, each of these steps that I have mentioned here, we will talk in details in our subsequent lectures. So, today, what did we learn? We learned about the definition. We learned about the principles, that is creation of sterile environment and genetic engineering. Then the different steps involved: isolation, cutting, where the use of enzymes, use of gel electrophoresis to separate out as also to create multiple copies, then how it is joined, how it is transferred, and how we produce the product. So this was the introductory, and subsequently we will learn about each of the steps in details.